Okay, so for our last system of the body, it sounds very weird to say that, given that we've only been together a short while, we are going to be talking about the reproductive system. And interestingly enough, it seems like each time we get to a new system, what we do is we look at the big picture, we say, okay, how does this system help us with maintaining our internal condition, maintaining homeostasis, and keeping us living and stuff like that. Then we got to zoom into the different organs that contribute to that system. Oftentimes we go down to the cellular level, check out what the cells do that give rise to the tissues, organs, and on up from there. And then kind of zoom out again. So, unique aspect of the reproductive system, though, will be the fact that this, of all the systems of the body, has the least to do with maintaining your body in terms of homeostasis, right? This system is about making other bodies. That's the whole idea with this system, okay? Now, we also did say that this system is dual function. So it's about reproduction, about making new organisms, but it's also part of the endocrine system. So we know that the gonad, the testes, and the ovaries are going to release sex hormones. Those hormones that are of the steroid class that are based on cholesterol and that have a function, for the most part, having to do with reproductive activities. And so they kind of tie in together. So we're going to look more into those functions of those reproductive hormones here, where we kind of skip over the endocrine system. Okay, so it's definitely unique in that it has very little to do with homeostasis. The other unique aspect of it is that it is different between males and females. If you don't know that yet, hopefully you'll be learning that in this class. Okay, so it's like two different systems in one, so it takes a little bit more going through, so I'm happy that we're able to start it today so that we can then finish up tomorrow and then go over any questions that we have, etc. So between the two, between male and female reproductive systems, we certainly have some shared functions. So primary functions, we'll find that these systems are going to be a series of glands and tubes that function to produce and nurture sex cells. These are also called gametes, okay? So that's going to be the sperm and the egg. So they're also going to work to transport them to the site of fertilization. So it's all about these gametes, creating them, supporting them in some way, and trying to get them together, okay? Now, that's a generalized function that we could use to describe both the male and the female, but we'll also find then that the female reproductive system is going to be about supporting the developing embryo and fetus as well. So that's going to be exclusive to the female reproductive system, so there's going to be a whole other kind of segment of the chapter that we're going to be talking about when we talk about development in that way. So in that way, we've kind of combined Chapter 19 and Chapter 20. Chapter 20 is development. But what I did is I kind of co-opted the information we needed to know from Chapter 20 and put it into this Chapter 19 lecture. Okay? So for reading and things like that, if you need to clarify some things about development and the embryo, the fetus, and things like that, you can look into Chapter 20. But for the most part, what you need to know about development we'll see in this lecture, Mind and Reproductive System. Okay, so given the fact that the female reproductive system is a bit more complex, we know that females are a bit more complex, right? We're going to transition into it then. We're going to start with the male reproductive system, which is a little bit simpler, and work our way up to female, okay? Okay, so the other interesting thing that we'll see as we kind of come full circle, if you will, is that when we initially started off, we were talking about, like in Chapter 3, we were talking about cells. So how cells do their job in order to stay living and how they will reproduce, right? So when we were introduced to cell reproduction way back when, what was that called? It started with an M. That's prophase, metaphase, anaphase, telophase. What? Yeah, mitosis, right? So cell division, where we are kind of making a Xerox copy of the original cell, and then we have two cells, two epithelial cells or two muscle cells or whatever, that's mitosis. So we start out with a cell that has, at least in a human cell, 46 chromosomes. So that's a full set of chromosomes for a human cell, and half of those chromosomes, 23 came from your dad, half came from your mom, so that gives us the 46. Okay? So that's for most every cell in your body. Now, we've seen some exceptions along the way. Red blood cells, mature ones, don't have a nucleus, so they don't have chromosomes. Skeletal muscle cells have multiple nuclei, so they're going to have more than 46 chromosomes, etc. But in general, a skin cell or a neuron or something like that has 23 plus 23 or 46 chromosomes. It's considered to be what's called a diploid cell, diploid, dip. The di part of it means two. It means you have two copies of those 23 chromosomes. Copy from your mom, copy from your dad. So that's 46. So the interesting thing that we're coming full circle with is that this system is the system that exemplifies our other type of cell division. We learned most details about mitosis way back when for repair and growth and stuff like that. Like if you get a cut, how we heal it over. But meiosis is the other type of cell division. And in meiosis, we take an original cell, and in a way, what we do is we tear it in half. And so what we get are not exactly as the same diploid cell we started with. Now we get half cells, or haploid cells, H-A-P-L-O-I-D. So we get two haploid cells, sometimes four haploid cells, each one with only 23 chromosomes. Okay? So the sperm and the egg cells that we produce are only going to have 23 chromosomes. And that's very important because when fertilization occurs, when, by chance, the sperm and egg come together, we want 23 from the dad, 23 from the mom, so we get up to 46 again. If we did not do meiosis, and we did mitosis, then we'd have a 46 diploid cell, coming together, and we'd have a 92, right? Like a quadloid cell or something like that. And you've likely heard of different types of genetic diseases and disorders where if you are missing part of a chromosome or you have an extra chromosome or something like that, it has some very negative effects in terms of development or something like that. Okay, so you can imagine if you had a full extra set, there would be problems. So the meiosis, that type of cell division, is what we're going to be talking about here. How it's done in male, how it's done in female, and then once we produce those gametes, how we support them, transport them, and stuff like that. Okay? So, if the main function for this system is to make new individuals, then if we are looking at the most important organ, how like we did with the digestive system, we've actually got to decide that the gonads, the testes, and the ovaries are going to be the most important organs, right? Most people, vagina and penis, definitely the most important organs, right? But if you don't have the gonads making eggs and sperm, you can go with do activities with those other organs all day and night, which is fine and fun and whatever, with your married mate, etc. But you're not going to have the system achieving its goal of making new individuals. So that's why the gonads are called the primary sex organs, because those are the ones where the sex cells, the gametes, are going to be produced. And you have to have those in order for the system to achieve its goal. Okay, so the primary sex organs are going to be the gonads. All of the other sex organs are called accessory sex organs. So a bit like we talked about with the digestive system. They're each important. They all have a role to play. But in this case, the primary ones are going to be the testes and the ovaries. So for the male, the testes are going to produce the sperm. It's also the site of the sex hormone production. So this is where the testosterone is going to be produced as well. And we'll talk about that coming up. Okay, so this view is a typical mid-sacral cut where we can see the different organs of the reproductive system. So you'll see when you're doing your exercises that we have models of this, both from like the torso model, we have this big blue or purple plaque where it has the same type of cut. And what you'll see is then, this is going to be our primary sex organ here. So we're going to have the testes down here, housed within the sac-like scrotum. And we'll talk about why that's important, that we have the gonads in the male outside of the main body cavity, as opposed to inside. So we have the testes. On top of the testes, we have the comma-shaped epididymis that we see here in red. From there, we have two that's called the vas deferens. It's also called the ductus deferens. You can use either name. And so the sperm are going to be created in the testes. they will be moved to the epididymis. From the epididymis, they'll go through the vas deferens or the ductus deferens, and they're going to be coming from each testis, from each epididymis, so we're going to have one of these on either side. We can see how it goes around the lateral side of the bladder, right, which we just talked about. And then, as it comes around here, it's going to pass by one of three different accessory glands we have in the male. The first one, right here, are called the seminal vesicles. So there's two seminal vesicles, okay? And as this tube comes by the seminal vesicles, we then call it the ejaculatory duct. Each of these glands are going to contribute some secretion that will aid and support the sperm in some particular way. And we'll talk about that coming up too. Okay, so we go from the vas deferens to the ejaculatory duct, and at this point right here, we see the ejaculatory duct coming in and meeting with the urethra. So we see the urethra coming from the bladder again. We see it passing through the prostate. The prostate is the second of the three accessory glands. Okay? So the prostate is going to dump in its secretions into the urethra there. And then that urethra is going to continue on. And in the male, we actually have three different sections to the urethra. The section that goes to the prostate is very clearly called the prostatic urethra. Then there is this little section right here, which is called the membranous urethra. That's where it's kind of passing out of the body cavity. And then the last and longest section is going to be either called the penile urethra or the spongy urethra. And the reason that we call it the spongy urethra is that there are three different areas of spongy tissue, or basically erectile tissue, in the penis. So we have two of them that are up here on the dorsal surface, and these are called, each one's called the corpus cavernosum. Okay? There's two of those. We can only see one, so there's one on either side. But the third one is called the corpus spongiosum, and this erectile tissue actually encircles the urethra. So all this you kind of see in that brown color is going to be the corpus spongiosum, and you see on the distal end, it kind of expands to become what's called the glans penis, or the head. And this will, in some people, be covered by the prepuce, or basically the foreskin. Okay? So the reason that we call this last section the spongy urethra that's passing through the corpus spongiosum. So that's the third section: prostatic urethra, membranous urethra, and then spongy urethra. Okay, so we can see now, these are all the different tubes and glands, etc., that are going to be part of the accessory structures of the male reproductive system. You can also see, important for males, right? As you get older, you hear about things like prostate cancer and testicular cancer and stuff like that. So it's important to have regular inspections, if you will, digital inspections. So for guys, they're supposed to feel the surface of their testes inside of the scrotum to feel if there are lumps, just like a woman will do, inspect for lumps within her breast tissue, etc. For the prostate, you're supposed to go to a doctor instead. Don't go checking your own prostate. But what we can see from this mid-sagittal cut, though, is here's the last section of the colon, right? So we see the descending colon coming down, the sigmoid colon, and then the rectum here. And you can see that the rectum and the anal canal is very close to where the prostate is. So that's why, in terms of examining the prostate, you go and the doctor puts the glove on, and he basically goes through there in order to feel the surface of the prostate again to see if there's any lumps or to see if it's too large or something like that. Okay? So you see that in movies and things like that, you're like, why does the doctor put his finger up the guy's butt? I don't get it. Right? It's because of the proximity to the prostate. So you don't want to go up the other way and go, examining the prostate that way. Okay. So what we're going to do is, much like with the digestive system and stuff like that, we'll do a tour. So we're going to start where the sperm created. We'll talk about meiosis and how the sperm come to be, and then we'll start moving our way through these different ducts and glands and talk about how they contribute to the reproductive function. Okay, so in the testes themselves are a bit like kidneys in that when we look at the surface of them, they seem pretty solid. But when we got into the kidneys, we found out that the kidneys are in fact made up of all these tubules winding next to one another, etc. There's a lot of surface area in there for these tubules to be able to do what they have to do. And the testes are very similar to that. So inside of the testes, we have what are called seminiferous tubules. So if we take a look at it, and you'll see this figure in the lab as well, right? Here's the testes right here, and this just gives us a little look into all the different tubules. It's actually organized into lobules and stuff like that. It's made up of all these seminiferous tubules. And if we zoom up on this little section right in here, what we're seeing is a cross-section of one of these seminiferous tubules. And in the cross-section of the seminiferous tubule, you can see that you have all of these stratified cells. So when we have layers of cells, like epithelial cells, we say that it's stratified, right? So these are stratified cells here too. But it's very interesting because those that are along the outer part in the seminiferous tubules, these are what are called germinal epithelial cells. In a way, these are kind of like stem cells. Remember when the skin is down at the very bottom, we have active cells that were dividing, and then they would kind of go and migrate upwards, and they would kind of change, etc. In a way, that's what's happening here too. So out at the periphery out here, we have the germinal epithelial cells. These are diploid cells, 46 chromosomes like most other cells in your body, okay? But these are the cells that are constantly kind of undergoing mitosis, and then some of those will undergo meiosis. We'll tear them in half, if you will, in order to make the haploid cells, in order to make the sex cells. But if we were, taking those germinal epithelial cells, and all we were doing is meiosis and tearing them in half, very short order, we would run out of germinal epithelial cells, and we wouldn't be able to make more sperm cells. So we've got mitosis that are making sure that we constantly have a supply of the germinal epithelial cells, these diploid cells around the periphery. And then those cells will undergo meiosis and end up making our gametes. And as we move further into the inside of these seminiferous tubules, we actually have developing stages of sperm. So we start out with a very generic looking round cell with a nucleus in the center. And as we'll see in short order, those cells, as they go through undergo meiosis, they will change, right? There's a very unique sperm shell shape. It's unlike any other cell in the body. And so they're going to undergo changes that will then eventually end up with mature sperm cells right here in the lumen, the space in the center there. You can see those little tails extending into the lumen in the very inside. So you're going to see a developmental pathway for these developing sperm cells. Now, before we leave this picture, I also want to highlight the fact that, while all these cells here are cells that are going to develop into sperm cells, or these cells that are supporting the developing sperm cells, we do have some cells that are outside of the seminiferous tubules, like here, and here, and here. See those little islands of cells that are outside? Those ones are called interstitial cells, right? Inter means between. And so the interstitial cells that we see outside of the seminiferous tubules, they have different functions. Inside the seminiferous tubules, it's all about making sperm. Outside of the seminiferous tubules, it's doing the other function of the testes, and that's to make the sex hormones, testosterone. So those interstitial cells are the cells that actually make the testosterone. So it's different little populations of cells outside of the seminiferous tubules that are the ones that are producing testosterone that we know come from the testes. Okay? Okay. So we said that the goal of these cells inside of the seminiferous tubule is to make sperm cells like we see here. So here's our very unique sperm cell. It's got a head to it, which contains the 23 chromosomes, at least when we're done dividing. It's got a mid-piece to it right here, which is kind of like the motor. Remember how we talked about, the all actin and myosin and sliding filaments and stuff like that that can produce movement? Similar types of mechanisms occur there in the mid-piece. But what will happen here is those filaments that they're moving will actually rotate the tail. So the tail is also called the flagellum. We know that a lot of cells, we have cilia that will move, like the lining of the trachea and stuff like that. They're supposed to move the mucus along. So cilia will tend to move things past the cell. This is one of the few cells in the body where we have just one. It's much longer, and when it moves, it actually moves the cell itself as opposed to moving other things past it. So this is a flagellum, or the tail, and the midpiece has a lot of mitochondria with it for producing that action with the sliding filaments and stuff like that. And then the flagellum can move so that this whole cell can move on its own. Now the other critical aspect of this cell is up here. You see that hat that it's wearing? That hat is called an astrosome. Now we know that ultimately the goal of the system is to get the two gametes together, the sperm and the egg together. But when the sperm contacts the egg, ultimately the egg is going to be kind of the house, if you will. It's not only going to have the 23 chromosomes from the mom, but it's going to, it's like if you get together with somebody and you move in with them, they have a bigger house, so you move into their house, right? This sperm cell is very, very streamlined, right? So it doesn't have a lot of room. So it's just basically 23 chromosomes in a head, and then a tail to move it around. And so when it gets to the egg, it's going to contact the surface of the egg, and we need to get those 23 chromosomes contained in the head inside of the egg. And that astrosome is basically a bag of enzymes. Okay? And so the enzymes contained within that bag, when they contact the surface of the egg, those enzymes will work like digestive enzymes in breaking down the outer layers of the egg, and it'll allow the head of the sperm to get inside of the egg so that we can get the 23 chromosomes from the sperm together with the 23 chromosomes from the egg. Okay? So we'll see that coming later when we start talking about development. But the astrosome is a bag of enzymes that will allow the sperm to enter the egg. Okay. So the first thing that we need to talk about, as we're still focused in the testes, okay, is this process of what's called spermatogenesis. Now, generically called gametogenesis, or basically the creation of gametes. But these gametes, the male, are called sperm, so we call it spermatogenesis. So what we've done here is we've zoomed in on the wall of the seminiferous tubule. So you might think that, you know, we've got like this big circle like this, okay? And we're zoomed in on, like we said, just the wall right here. So up here would be like the lumen, the open area, where the fluid would be and stuff like that. So this would be, up here would be the inside of here, and this would be the lumen. The lumen is going to be the open area inside of any hollow organ, like the inside of the stomach or the inside of the ureter or something like that. Okay, so we're zoomed in on the wall of one of these seminiferous tubules. Out here is where we see those diploid germ cells, right? These are the ones that are going to undergo meiosis. Now, this earliest stage, these are the most immature, if you will, sperm cells. This earliest stage, these are called spermatogonia. An individual one is called spermatogonium, with the M on the end, but collectively they're called spermatogonia. And we can see that they have 46 chromosomes. They look like a generic cell. Now, when they start their process of meiosis, of this kind of tearing in half, the way that it works is it's a bit like mitosis, except it's got two rounds of prophase, metaphase, anaphase, and telophase. Okay? So same thing in terms of like lining up chromosomes, meet in the middle, they move away from each other, then we tear the cells in half. We're going to go through two rounds of that. So if we think about it, when we did one round of that, we took one cell, we divided it, and at telophase, we tore it into two cells. So we're going to do that twice. That means these two cells are also going to end up dividing into two. So we go from 1 to 2, then 2 to 4. And we have two rounds of prophase, metaphase, anaphase, telophase. Okay? So, what we're going to see here is between spermatogonia cells and the primary spermatocytes, we're starting to get a little bit smaller. And when we transition between primary spermatocytes and secondary, notice we go from one cell to two cells, and we go from 46 chromosomes to 23 chromosomes. Okay? Now, if you remember that much like mitosis, we have this interphase, this between phase, where we are undergoing duplication, right? Where we are basically taking the genetic material and running it like a copy machine. We're like, okay, chromosome 1, we need 2 copies of that. Chromosome 2, we need 2 copies of that, etc. Okay? So when we start out with this process, we actually have 2 copies of all 46 chromosomes. And when we tear it in half, this first time, between primary and secondary spermatocytes, what's going to happen here is that those 2 cells that we produce, the secondary spermatocytes, only have half of the genetic material, okay? So it's kind of like taking your textbook, and you made a copy of the textbook, so you have 2 copies of the textbook, 2 full copies, and then what you do is you end up tearing those textbooks in half, okay? So like these 2 cells over here or this cell over here is going to get, in essence, 2 copies of chapter 10 through 20. And this cell over here has 2 copies of chapters 1 through 9 or something like that. Okay? So that's what we get when we get to the secondary spermatocytes. They only have half the genetic material, 23 chromosomes. Okay? But they have 2 copies of the 23, of the single 23. So when it goes through the second division, between secondary spermatocytes and spermatids, now we go from 2 to 4 cells, but notice that each of these cells still has 23 chromosomes. Because the secondary spermatocyte has 2 copies of the second half of the book. So when it divides, each of those cells it divides into to become spermatids still have, like I said, half of the cookbook. They have 23 chromosomes in them. Okay? So, at the end of this process, what we started out with was 1 spermatozoon that has 46 chromosomes that have been duplicated. At the end of this process, we end up with 4 mature sperm cells, 1, 2, 3, 4. Each has 23 chromosomes, and each, through this process, has become specialized in terms of the cell shape, too. You notice that each of these steps along the way, it becomes less and less like a generic cell, and more streamlined to be able to move through the aquatic medium, like in the movement of the testis or the tube that we're going to talk about, etc. Okay? So we started out with 1 cell that was diploid. We ended up with 4 haploid cells. So that's very good because this process that we're talking about for spermatogenesis is kind of an on-demand process. It starts at reproductive maturity, okay? So when the sex hormones get up and going, this process gets up and going, and whatever your young teens or whatever time it starts these days, when you're 10 or something like that, 11, then in essence, we're going to be producing these sperm on a continual basis, okay? And as ejaculation, there are multiple millions of sperm that are going to be in a single ejaculation. And so you can imagine, if we're needing to release that many cells on somewhat of a regular basis, we're going to have to produce a lot of cells. So this way, we start out with 1 cell, we end up with 4 every time we go through this. So we're able to kind of amplify the number of cells that we can make. So we have this continual kind of volume of sperm that are ready to go. Okay. So now that we know where the sperm are coming from in the testis and the seminiferous tubule, we have to talk about where they go from there and how these other accessory organs help out. Okay? Now this list that you see here, the reason that the spelling is a little bit weird is that we're talking plural. Okay? So there's 1 epididymis on top of each testis. Okay? So together they're called epididymes. And then coming from there, we have 2 vasta deferentia. Each one's called a vas deferens. Okay? Then we have 2 ejaculatory ducts as they're going by the seminal vesicles. Then we have 1 urethra. Okay? There's 1 prostate gland surrounding the section of the urethra, and there are 2 bulbourethral glands. That's the 3rd gland that we hadn't mentioned previously. Okay. So, that common-shaped epididymis, why is it there? In essence, all the production of the sperm are occurring down in the seminiferous tubules, but as we're making the sperm, we need to kind of offload them and store them somewhere. That occurs in the epididymis. So the epididymis is the final site of sperm maturation, and it's also a site of sperm storage. So while they're produced out of the testes, they're going to be stored in the epididymis. Okay? Okay, so from the epididymis, they're going to be transported through the vas deferens, the tubes that are leading into the body. So at that point, the vas deferens sounds like the esophagus or the ureters or whatever. It's just about transport. Nothing's going to be added to it, not doing anything special. We're just moving it along. Okay? Okay, so those vas deferens, or vasa deferentia, go around the sides of the bladder, and they start passing by the 2 seminal vesicles that are on the backside of the bladder. So what do the seminal vesicles do? So this process where we're moving the sperm inside, okay, is called emission. It's emission when we're moving it from the testis to the epididymis, to the vas deferens, to the ejaculatory duct, to the urethra, etc. That's called emission. It's ejaculation when it actually leaves the body, okay? So during emission, seminal vesicles are going to secrete alkaline fluid. Now, the importance of that alkaline fluid is a few different things. Number one, remember that the sperm are going into the urethra. What else goes in the urethra that we learned? Urine, right? Urine tends to be pretty acidic. So when you're looking at, for instance, the urinalysis and stuff like that, you're going to use nitric, right? You did that system yesterday, urinary system and thing. Okay, that reminds me, okay, with the urinary system lab, there's two parts of that lab. One is the urinary anatomy, the other is urinary physiology, okay, or basically urinalysis. So this is one of the few times when you go into those instructions where it's telling you, okay, look at this figure, like what and stuff like that, this is what's going to be on the quiz. It's one of the few times that there's more than one of those pages. So when you click like on the labeling exercise, or you're going through stuff like that, when you get to the bottom of the page, there's a link that says next page. You have to click on the link that says next page to take you to the next page where it actually goes through, for instance, the aspect of your analysis and stuff like that. It talks about the things that you would normally find in urine, etc. Should you find glucose in there? No, not normally. So should you find what's the concentration of it, what's it called, specific gravity, what's the pH of it, etc. Okay. So my point right there is that when you're doing the lab tonight, don't forget to do the urinary anatomy part and the urinalysis part. It's like 3 different pages. Because when you get to the quiz, guaranteed there's going to be questions on urinary anatomy and urinalysis. If you forget to do those other pages, you're going to be like, what? I've never seen this urinalysis test here before. What's it asking about? Okay. Anyways, the important thing here is that the urine and the sperm are going into the same tube. And oftentimes, the urine is acidic. Okay? It helps to retard bacterial growth and things like that. So oftentimes, the pH of a urine sample might be like 6. Could be down to like 4 or something like that. And so, an acidic environment is hazardous to a lot of different cells, bacterial cells as well as our own. Okay. So the sperm being dumped into this tube, where there's this very somewhat acidic fluid that goes through regularly. So the secretion from the seminal vesicle is alkaline. Okay? That means that it helps to neutralize the acidic environment inside the urethra. As well, the vagina is also acidic. And so while in the vagina we're looking to, again, retard bacterial growth and things like that, it could also kill off sperm cells. And so the sperm are looking to try to be protected in some way. And so the secretion from the seminal vesicle helps to neutralize the acids in the urethra as well as in the vagina where it's potentially going to be positive. Okay? So that's helpful. The other thing that is helpful about the seminal vesicle fluid is that it contains fructose. What's fructose? It's sugar, right? So remember that these cells are very active cells, right? They have a whole humidity, it's got a lot of mitochondria in there, we're going to be moving a lot, etc. And so these cells are going to need energy, so why not just bathe them in energy molecules that they can use in order to fuel their activities? Okay? So there's sugar in there, that's important as well. And there are some prostaglandins. Prostaglandins are chemical messengers that can stimulate the female reproductive tract to actually contract and help to bring the sperm further into the female reproductive tract, getting them closer to the potential site of fertilization. Okay? The other thing that is kind of interesting, that's not listed on here, is that there are some proteins that are secreted by the seminal vesicle that cause, in essence, kind of coagulation. So we talked about, clotting and stuff like that with blood, and how there are proteins that can make things become stuck together. In fact, the seminal vesicle also has proteins that can make semen become very viscous. And the idea there is, once it's deposited in the female reproductive tract, it's going to be important that these sperms stay there so that they can have a chance to get and migrate up to where the eggs are going to be. Okay? So by having these, if you will, kind of clotting factors or coagulation proteins in there, if it's deposited there, it helps it to stay in the vagina, stay in the female reproductive system, giving it a better chance of staying and getting to the site of fertilization. So all these things are looking to help the sperm do their potential function of getting to the site of fertilization and potentially make a new individual. Okay? Okay, so seminal vesicle secretion can help in various ways. All right, so we get past the seminal vesicle, go into the ejaculatory duct, and now we're going to jump into the urethra and then actually into the prostate gland. So we said we have one prostate gland. So the question is, what is the prostate having to do with the semen? Remember, the semen is going to be the sperm cells that are coming out from the epididymous and the tubules, plus the secretions from these different glands. So the prostate gland has a couple different things it's going to contribute. Number one is alkaline again. Again, to help neutralize the acidic viruses that it's going to be passing through. And also, it also has a different protein in it. So remember when we talked about the the seminal vesicles having kind of a coagulation protein? Well, you can imagine on a certain level, okay, that's good. We can get the sperm to stay in the female reproductive tract. But imagine, right, these sperm have to be able to swim. They actually have to swim from the vagina through the opening of the uterus called cervix. They have to go through the uterus. They have to go into the uterus tubes called fallopian tubes in order to meet the egg that's been released by the ovaries. Okay? There's a long way that they have to go on their own swimming. And so, if we stick the wall inside of the female reproductive tract in the vagina, that's great to get them to stay there. But how are they going to get up and get out of there or to get up to the site of fertilization when the egg comes? Well, the protein that's secreted by the prostate gland actually reliquefies the semen. So for about the first 5 to 10 minutes or so, that protein from the seminal vesicle makes it really viscous. And then after a while, kind of like the whole fibrin and plastin thing, the protein from the prostate gland actually reliquefies it so that the sperm can continue their way up. So it's pretty interesting, like I said, a bit like the whole body cascade and the plasma dissolving in the clot that we saw in the cardiovascular system. Okay, so we made it to the prostate gland, and we made it past the seminal vesicle. Now we're going to the urethra for passing by the urethral gland. These are also sometimes called the Cowper's glands by Dr. Rob Cowper or whoever that found them originally. And these also produce a secretion, but basically they put out an alkaline mucus. Now this mucus, while some of it will come out and contribute to what we call the semen, that volume that's being emitted, most of that mucus is going to come out prior to ejaculation. It's going to be there for lubrication. So that secretion that we have there is also going to be important, but it's contributing less to the actual semen itself. So the bulk of the semen is really going to come more from the seminal vesicle and the prostate gland itself. Okay? But it's a third gland that is going to be helping the reproductive function in some way. Okay. So like we said, semen is going to be a combination of the sperm cells, approximately 120 million per milliliter. You know how much a typical ejaculate is? It's like 5 milliliters. So you scale that up like 600 million cells in one ejaculation. So you can imagine it's pretty important that down the centimeter tubule, we're doing that kind of amplification where we're starting out with 1 for getting to 4. And there's millions and millions, hundreds of millions of these cells that are being released on a regular basis, depending on if you're married or not. Okay, so the sperm needs to finish its maturity, like in the epididymous and stuff like that, and they also become more active in a process called capacitation when they're deposited in the female reproductive tract. So whereas they were kind of slightly swimming before, they become much more vigorous and active in their swimming as they get deposited within the female reproductive tract. Okay, so what we've done right there, we've done a tour through the internal accessory organs of the male reproductive system. So now we can talk about the external reproductive organs. So we said that the testes exist within a sac that's called scrotum. So, the importance of that is that the spermatogenesis that we talked about, that whole meiotic division, etc., in the sperm, it's important that that actually occurs at a temperature that is below body temperature. So body temperature typically it's 98.6 degrees, but that's actually the temperature at what we call the shell. So if you're measuring one of those infrared thermometers or something like that, or even one of those on your tongue or something, you're measuring that temperature at the surface where we're actually radiating heat. So if you're going to core body temperature, it's actually more like 100 degrees Fahrenheit, okay? And 100 degrees Fahrenheit, for the different enzymatic reactions that actually make mitosis occur, it's too hot, okay? The enzymes that we have, like digestive enzymes or whatever, have to have a specific pH, a specific temperature, etc., okay? And for those molecules for mitogenesis, it's too hot inside the body cavity. So, we have these testes that actually descend when you're a baby. They descend into the scrotum. And so, then the scrotum is able to keep those testes at more of an optimal temperature for spermatogenesis. Okay? So, that's the importance of the scrotum, is it allows spermatogenesis to occur at a more optimal body temperature. Now, the interesting thing with that, if you think about it, we don't go through all of these structures, but there are various muscles that are associated with the scrotum. And so what those will do, it's called the Darder's muscle, stuff like that, is those will constrict or dilate and cause the testes to raise or to drop, depending on body temperature, right? So if you talk about something being very cold and there's shrinkage, okay, in essence, the testes, right, what's happening is those muscles contract and bring the testes close to the body to keep them warm. Keep them close to where the body temperature is higher because it's colder outside. Now, on the other side of the coin, it's warm and stuff like that. It's too warm here, right? We're radiating heat, we're evaporating heat, sweat and stuff like that. So it's extra warm here. So we want those testes to be even further from the hot body. And so in that case, the muscles in the scrotum will relax and the testes will descend instead. So there's a very regular process of kind of raising and lowering the testes in order to maintain that body temperature like we talked about, or at least the optimal temperature, lowering the body temperature. One time when I was watching, Discovery Channel, they have all these various shows on topic. They have a show that has to do with reproductive systems and stuff like that. And then they had this one story about these Scandinavian researchers that were looking into that fact that spermatogenesis needs to occur at body temperature, or at a temperature lower than body temperature. And what they did wasn't exactly very good scientific method, right? So step one, replication and all that kind of thing, right? So they did this experiment on themselves anyway, and they're male. And what they did is, perhaps you've seen like hoppers and one that have a seat that has a lobe in there where you can actually, if you have to go to the bathroom, you can't get to the toilet, you can actually see those that cutout area, and it has a basin underneath it. So what they did is they filled the basin with water that was maintained at 100 degrees Fahrenheit. And then what they did is they sat with their testes in the 100 degree water for substantial periods of time, and then measured the amount of sperm that they were actually producing. And what they saw is that at the time, their sperm count basically went down, to almost nothing. And then after a period of days, it actually came up again. So that's why, people are having difficulties conceiving hot and stuff like that. That's one of the first things they ask. So what's going on with the guy? Well, you go hot stuff regularly? Oh, hot stuff are regularly above 100 degrees, right? Like whatever, 103, 105, something like that. So that can increase sperm count. Are you wearing tidy whities as opposed to boxers? Well, that's going to keep the testes right close to the body, and that may make you warm on a regular basis, etc. So it's interesting to see how much the body is working to maintain those things, even when we're not aware of it. Okay, so function of the scrotum. So the penis. So the penis is supposed to be able to deposit our sperm cells within the female reproductive tract. Now, like I said, also when we can bang urine to the outside, but within the penis, like we saw, we've got 3 different columns of erectile tissue. Now, we saw it in the mid-sexual cut. If you were to see it more in a cross-section, which I don't particularly like to see, but I'll show you, before vegetation, okay? It basically has 3 different areas to it, sounds like this, okay? Now, inside of the upper 2, These upper two up here are basically corpora cavernosa up there. Okay? So those are corpora cavernosa up at the top right there, right? Oops, sorry. Here and here. So the urethra is not going through there. The urethra goes through down here. So the bottom one is the corpora spongiosum. So in here, that's where the urethra is. So this gives you the idea that there are three different areas of erectile tissue. Two corpora cavernosa, one here, one there, and then corpora spongiosum around there. We said that this one, corpora spongiosum, exposes, kind of expands to be the land of the penis or the head. And the way that this erectile tissue works is that it will, like capillary beds and things like that, it can fill with blood. But the interesting thing about it, when I was drawing the corpora cavernosa up here, is that there are a series of arteries and veins that go into these areas of erectile tissue. And like we saw in the cardiovascular system, oftentimes there will be an artery right next to a vein. And in this case, what happens is, when the erection is needed, what happens is we actually dilate the arteries. And so arteries go away from the heart, right? And so they're going to drop blood into the erectile tissue. Now, because the artery and vein are right next to one another, when the artery expands during dilation like that, then what happens is it presses up against the vein right next to it. And so the blood can go in. But the blood can't take it because the vein is blocked because the artery has expanded so much. So we have parasympathetic stimulation that goes to the artery that says it should dilate. The vein now compressed, so that maintains the blood within the erectile tissue. Okay? So it maintains the erection. Now afterwards, what happens is, right, we want blood to leave the erectile tissue. So now we're going to constrict that artery. And the vein is now not going to be compressed anymore. It's going to give a venue for that blood to leave. And now the erection is going to go away because the blood is going to be drained out. So it's an interesting system. And rather than like the rest of our arteries are getting, when we want them to constrict, we use sympathetic stimulation and they go to constrict. When we want them to dilate, we just stop stimulating them and they naturally get bigger. With this system, it's a little bit different. Parasympathetic tells us to dilate, and then sympathetic will tell us to constrict. And it affects the artery, affects the vein right next to it. So it's very interesting. Okay. So, like I said, it's going to be parasympathetic impulses that allow the erection, that allow the blood to go into the arteries and compress the vein, and then it's going to be sympathetic that's going to constrict those again. So the emission is going to move the sperm from where they've been stored at the end through these different tubes, like that difference in the ejaculatory duct and the urethra. The glands along the way are put in their secretion, and then all of that is going to be ejaculated, forced to the outside, and deposited into the female reproductive tract. Okay? So we said, after ejaculation, sympathetic and constrict the arteries, allow that blood to drain out. Okay. Now, for that, we're kind of saying there. So what we did is talk about how we generate sperm, how we move sperm, and package them with the secretions that are going to help them, and they get to the outside. We have to go to the other side of the point, talk about the female reproductive system in order to understand where they're going to potentially lead up with the egg. Okay, so that'll be coming as we finish up female. But before we get out of the male reproductive system, we want to talk about the hormones that are involved as well. Okay, so if we think back to when we talked about resistance first, we had this big control axis up here where the hypothalamus of the brain of the thymus on communicates with the pituitary gland. The pituitary gland releases a bunch of trophic hormones that go down and tell a bunch of other glands what to do. Okay, it's all about thyroid gland, hormones, etc. Okay. In the gonad, in the reproductive system, same thing happens. The anterior pituitary puts out two hormones that go down to the testes and tell us what to do. They tell us when to produce sperm cells, and they tell those interstitial cells when to produce testosterone. Okay? So if we look at it, we're going to have a releasing hormone coming from the hypothalamus, and it's going to tell the pituitary to put out these two hormones. One called luteinizing hormone, we abbreviate LH. The other one called stimulating hormone, and we abbreviate that FSH. Okay? So what do those do? If we look at it, testosterone is going to be the only hormone. It's going to be like the primary, let's put it that way. Male hormone is released by the testes. So only one of those hormones coming from the pituitary gland is going to control that testosterone release. So when we look at it, it's coming up. We're going to find one of those is responsible for testosterone release. The other one is going to be controlling the other functions of the testes and producing sperm cells. Now, testosterone that comes out, interestingly enough, the testosterone that comes out has a reproductive function. It increases the activity of sperm production, and it also helps to maintain the secondary sexual characteristics. Now, the primary sexual characteristics are what gonads have, whether you have a vagina or penis, etc. Okay? But the secondary sexual characteristics are going to be those body characteristics that also indicate whether you're male or female. So men, for sex you're men, maybe you have deeper voice, you have more of the terminal body hair in various places like on your chest, or your face, etc. Big things on the skin, and increased musculature of the upper body. Those are stereotypically male characteristics that are maintained by testosterone. Okay? And people that are taking steroids or taking testosterone exogenously, that's what they're trying to get. They're trying to get more of those types of secondary sexual characteristics, more body musculature, etc. Okay. And then you have more hair. There's important side effects, and one side effect among many is that testes tend to shrink. Because, right, the testes are normally putting out testosterone and maintaining those things. Now you're injecting this testosterone, okay? And the testes are like, well, great, I'm not going to do this. I'm sure I'm going to make, sperm cells like that, but the whole section of my job got knocked out. So we don't need to be as big anymore. We don't get our body response, so there's a lot less demand. Things get bigger, and there's less demand, things shrink. And so the testes tend to get very small, people that are injecting lots of testosterone. Okay, so testosterone also maintains secondary sexual characteristics. Alright, so what we do with testosterone levels are kind of interesting. There's a big peak before you're born, okay, that will cause the accessory organs of the male reproductive system to form. It's kind of like everybody starts out early on, kind of as a generic female reproductive system. But when you have testes, as opposed to the testes cell, as opposed to the ovaries, they'll release testosterone in utero. And that stimulation will cause the different producing glands, etc., to become male as opposed to female. Okay? So you think about before you're born, that helps form the male reproductive system, and then it goes very low until you reach sexual maturity or whatever, 10, 11, then testosterone levels go high again and remain for the rest of your life. So like we talked about before, there's very serious to it, right? Like a monthly cycle or a daily cycle. This is like 2 major periods. One cycle before you're born to get everything going, and then you reach reproductive maturity, and they stay high basically for the rest of your life if you're a male. Okay. So, like I said, what are these hormones that are coming from the pituitary? What does that do in terms of controlling the testes? So we have luteinizing hormone, LH, follicle-stimulating hormone, FSH coming down. Follicle-stimulating hormone is mainly control of producing the sperm cells. So it stimulates the spermatogenesis. Okay? So that's the main role, is that it encourages the embryo to produce more sperm cells. Luteinizing hormone in males is also called interstitial cell stimulating hormone. And we know the interstitial cells are those that are releasing testosterone. So that hormone, luteinizing hormone, also called interstitial cell stimulating hormone, is responsible for the other function of the testes, controlling and releasing testosterone. Okay? So we see that the pituitary gland is really in control of what's going on in the testes. It's a management level type gland. We'll see that again with the female reproductive systems too. Okay. So that is a male reproductive system. Question on that? Seems relatively simple. Okay. So, a bit like this is for reproductive system, one critical thing to know is we have all these different organisms. Why? What does that mean? What does the medical do? Different prostate like different reproductive? What the activities do for testing, etc. Okay? So that's the big thing. Doing training somewhat to help things how things work together, right? Maybe testing, they go to the end, they go to that friends, etc. So those are good exercises to do. Okay. So what we're going to do is do an introduction to the female reproductive system. Now we'll jump ahead as well, so that we can kind of start in the middle at the same time. So for the female reproductive system, similar functions producing gametes, in this case K, right? For oocytes. Also, very important cell inside fertilization, again, the same thing as the male reproductive system. But when we look here, it says produce sex hormones. In this case, it produces female sex hormones, so it's the male. And then this, we see here, and that's there. We're going to use this as female reproductive system, right? Maintaining development embryo fetus and finding birth. So that will make things a little bit more flexible. We're going to talk about that too. Okay, so stereotypical that's about we saw in the male, excuse me. So here we see the gonad again. So in this case, we have the ovaries that are located inside of the body cavity. So that whole temperature thing is not common for oogenesis, and we'll find out why in a little bit. So we've got the ovaries there. And associated with ovaries are finger-like structures that are called fimbriae. And that's like actually blood, okay? Because the ovaries are where the eggs are produced and where they're released on the monthly cycle. So in general, there's going to be one egg that is released from one or the other ovary. And when it's released, it actually bursts through the cortex of the surface of the ovary. And for a very period of time, it's actually in the cell cavity, but it's caught by the fimbriae. So it's really just like protecting blood. And in fact, the fimbriae, they have smooth muscles, so they do this kind of sweeping motion to help gather it in. And in fact, there are affiliated cells in it, so they're going to begin to draw you fluid, drawing an egg, which are releasing the surface of the ovary. So the fimbriae collected, and then the egg is going to be transported into the uterine tube. This is also called the fallopian tube. And the fallopian tube comes down to the uterus, like we see here. And the uterus comes down and has an opening called the cervix, and the cervix opens down into the vagina. Now remember in this case that we're going to have separate urinary reproductive systems. Okay? So we have vagina more posteriorly. On the anterior side, we have the urethra, penetrating bladder, and anterior to that, we have the clitoris. So the clitoris is actually analog to the penis. It's actually erectile tissue, etc. And so all of these are enclosed within the labia. So we have the labia minor on the inside, and then the labia major on the outside. Okay? So what we're going to be looking at is, again, we're going to start with the ovaries and talk about how many eggs they produce. And then we're going to go on the pathway with these other organs and talk about their physical role. Okay. So we start out with primary sex organs. So primary sex organs are always using gonads because that's where the gametes are produced. Okay, and we're going to have both external and internal sex organs. So now with the ovaries are similar in a way, but also quite different. So with the ovaries, we've got an inner area called the medulla, and an outer area is out of cortex. And it's out of the cortex where all the important stuff's going on. It's a bit like the kidneys, right? The kidneys where all the nephrons were. And so here we're looking at the ovaries. It's the cortex that is going to be very important. And once I actually get up to this figure, and I'll come back to the slide. Okay? So here the ovary is oval shaped, just like the testes are. Okay? So this is the ovary here. The medulla would be on the inside and then here, and then the cortex would be on the outside. Now, what we're going to find is that you're going to do reference to things called follicle all the time. It's a little bit confusing to see up here, follicle maturation, et cetera. What? In the kidney, but that's in the kidney. When we have glomerulus and the glomerular capsule, those two things together, it's called follicle. It's like a package. Those two things together, it's everything. Okay? In an ovary, it's a bit the same. Okay? When we look at these follicles here, what we see is in the center, we have an oocyte, that's the egg. But then surrounding the oocyte, we have all of these little pink follicular cells, even in the small ones, even in the bigger ones, etc. Okay? So what we have is a follicle is equal to an oocyte egg in the center plus the surrounding follicular cells. So that's what a follicle is. Every time you see a follicle, it is possibly the egg. The egg is surrounded by these follicular cells, which have an important role in all cells. Okay? But the gamete is the oocyte in the core of it. So that's why you see follicles to the life. Okay, so the follicles are located out in the cortex of the ovary. That's where they are initially created, that's where they're stored, etc. So let's go back here then. Okay? So what we see is that we said that they have a nucleus on the inside and a cortex on the outside, and the cortex contains all those follicles. Follicles being the oocytes surrounding follicular cells. Now, with the follicles, it's a very interesting process of development for these follicles, these oocytes. So for the first cell, it was like a on-demand basis. So constantly, throughout the reproductive maturity, making sperm cells. Okay? But with the egg, all of the egg you are ever going to make, you made before you're born, and then they're stored in the ovary and released on a monthly basis. Okay? So there's no, okay, more eggs and stuff like that. We're going to release an egg next month to make some. Not anything like that. All the eggs you're ever released over your reproductive lifetime as a female were made before you were born. And they're stored in those two internal gonads. Okay? So what happens is that on a monthly basis, you're going to have some of those follicles have come to maturity. They've developed it through previous months. Usually they say, let's see, I think I screwed that recently. I have to check my notes. But usually they take a number of months for a set of follicles that are kind of stored on the shelf, if you will, in your ovary, to get a signal like, hey, you guys are going to be 5 by 10. You guys are winner. You might be the egg to be released in, a number of months now. Okay? So you got to start getting ready, getting bigger, stuff like that. Okay? So there's a set of these different follicles that are actually different stages of development for the chance they will be the ones to mature and release from the ovary during ovulation. Okay? All the rest of them are just sitting there waiting for their turn. So the ones that are sitting there in stages waiting for their turn are really small, are called primary follicles. Okay? And there's initially millions of those that are actually made. And they have a primary oocyte, the oocyte on the inside, and then they have the follicular cells that are surrounding them. Okay? Now what happens is that I usually have to talk about before. Well, it's going to start in order to make the oocyte, but it doesn't actually complete until way down the line, right? So we start the process of meiosis very early to make the oocyte, and then they're stored in the ovary, and they actually don't complete their meiotic divisions until number one, they're going to be actually one developmental process, and they're going to be released from the ovary during ovulation. And number two, they don't fully complete the whole second set of meiotic division until they're actually fertilized by sperm cell, right? So you have an egg that was created before you were born, right? So say 20-something years ago, these eggs were created. They start their meiotic division and then they stop. And they're stages just being stored in the cortex of the ovary. Okay? Now, some subsets of those will then continue to be developed on a regular basis, and then will start that meiotic division again. Okay? But at the time that they're releasing something like that, they don't finish the whole set of meiotic division until, if by chance, they get fertilized by an egg. So you can imagine, say, you don't get pregnant for the first time until you're like 30 or something like that. Those eggs, that one egg, has been waiting until it's meiotic division for like 30 and a half years, right? Maybe before you're born. So that's that part. Okay? So that's a long time to actually finish that meiotic division. In terms of sperm cells, like that, you're talking about a matter of hours or days or something like that, not years. Okay. So anyway, at puberty, you only have about 400,000 cells. So a lot of them have been reabsorbed or something like that. But when you look at it, and you count it up, right? From reproductive maturity, say like 10 years old, until menopause, when you stop ovulating, something like that, hormone levels drop. Say for number E, we'll say 50. So if you end at 50, that's like 40 years. 4 years, you got 12 months, you really one egg and a half per month. So that's 40 times 12, 480. So there's 405 eggs that are only going to get released and go to maturity throughout the reproductive lifetime. So we don't need that many. And so there's a lot of eggs that actually don't make it. So there's multiple hundreds of thousands that start out, but the long as they'll develop and the long as they break, this that 5 to 10 minutes ago, not all of them are going to make it. There's only going to get one that will win that race, and release across the surface of the ovary. Okay? So that's what we're going to talk about next time. We're going to look at the process of oogenesis, the meiotic division for the egg cell. We'll talk about the development of follicles, like we saw in this figure, where we go from the ones that are just being stored like that until we get to a mature one like this that's going to be released. And then once that egg is released, we'll start talking about how these other accessory organs are going to help it, and how potentially fertilization can occur. The sperm and egg, whatever happens then. Okay? So that's where we'll have to stop for now. But we should have no problem finishing up reproductive systems tomorrow. Go through questions, go through highlights and everything as well. All righty, that's do it for today. We're almost there. The end of the story. Or really, think about getting into the story. We're talking about pretty one. Look at that whole circle.
We're going to go to that, right?
Yeah.